important that I don't forget to do that. We only have half the class present, unfortunately. Um, One last thing. Does anybody remember how I can mute somebody? Because there's someone who called in, so I don't even see a name associated with
Yeah,
them.
it can be in your settings. You're able to do it. With moderator, you're going to be able to mute any of the participants.
Right, and normally I would be able to by, oh, there we go. Okay, attendee, okay. One tiny, yeah, for some reason for this caller, it doesn't allow me to do that. Everyone else, I have the control for them and I can, okay. All right, let's see, maybe the problem will resolve itself. Does anybody watch or used to watch the Norseman? The kind of Viking parody TV show, the Norseman? Ah, nobody? Oh, guys, you have some important viewing to do. The Norseman, it's hilarious. I just love it. You have to. You absolutely must watch this show. I don't have a line. I'm sure you do, but watching The Northman is going to enrich your life a lot more. Trust me, it's just hilarious. It's really a wonderful show. Next time I'm gonna get some quotes from the TV show just to kind of inspire you to watch it and fail your finals. But it's really really good. The reason I said it is because one of the characters says, "Well, you know what they say? 80% of problems solve themselves." And everyone's like, "Who says that? Nobody says that." And I find that now I'm quoting somebody called Orm, who's a giant loser, and he's hilarious. You must watch it. Of course, I can't do it justice by explaining it, but please do watch it and think of me. Okay. Now, today I'm going to be sharing my screen with you. And we're gonna be going over the ECM presentation. Some of the ECM stuff are things maybe you remember from earlier lecture, because we did kind of say how neurons and even just cells in general are not everything. Cells don't exist in a vacuum. So I really want you to really appreciate that message moving forward. And that's why we have an entire lecture dedicated to that. Okay? Now, as I share the screen, I want to ask, what are some of the things you remember about this ECM? And please, in order to do so, don't type, and just unmute yourselves and tell me, okay? What are some of the things you remember about the ECM, the extracellular matrix?
It's a significant portion of the cell, outside the cell.
Yeah, I don't know why I have the date as the 7th. Whatever. It's not the 7th, it's the 2nd. Probably I was thinking 2nd, 7th, they sounded the same. It's the space outside of cells. Yes, very good. What else? What do you imagine? Again, it doesn't have to be accurate. I don't expect you to know everything about the ECM. That's why we're learning about it now. But what do you imagine when someone says extracellular matrix, the space outside of cells?
A space that has a lot of collagen and glycoprotein that helps for cell communication and growth of the cell and migration as well.
Oh, so it's involved in a lot of important functions. So you mentioned something about some of the components, like collagen being an important component there, right? It's kind of a structural, important structural element of the ECM. But you also mentioned how it's important for cellular communication, for example, right? Or even growth, right? Very good. What else?
Yeah.
What else comes to mind? What else comes to mind?
This is kind of silly, but I usually think of it as like cotton candy or like insulation in a house. Like all of the pictures of the ECM and like the illustrated pictures in the textbooks, they all look very like full.
Yeah, yeah, it's almost like almost like a jello environment, or like an insulation cotton candy, that type of I guess what we're all trying to express is that there are these structural elements that are surrounding all the familiar things that we normally study, right? So all of these things are just outside the cell, and they're like insulation or cotton candy. Yeah, I guess, yeah. Because that's how it looks in drawings that include the ECM, because a lot of textbooks, when they're explaining something, they omit the ECM because it's going to just add to the background noise, and they want to have like a clear idea of the cell that they're talking about, which is completely understandable. But it just over time, having seen these pictures over and over again, we tend to think that there's nothing else in that space, which is of course wrong, because there's a lot of space that's taken up by these elements. Around 20% of the adult human brain is extracellular matrix. And one thing again that we've been mentioning is that how the brain is, prime real estate. We don't have unlimited space there. So anything that's there is super important. We don't just waste the precious space we have in the brain. And if 20% of it is extracellular matrix, it must be doing something very important. Mostly it's composed of proteoglycan and hyaluronan, and we're going to talk about the composition in greater detail. So, what are proteoglycans? Proteoglycans, as you can imagine from the name, it's proteoglycan. So it's a glycan molecule. It's kind of like a sugary molecule with a protein bit associated with it. Okay, so it's a complex macromolecule. It has these two main components, and it has a central core, and then we have something known as GAG chains. GAG chains are glycosaminoglycan chains, okay? And these are they use these link proteins, mainly N-link proteins, but can also be O-link proteins, O-link entities, oligosaccharides. Sorry, I keep on saying proteins. And these are covalently linked. So what I want you to get out of this is that I want you to remember that we're talking about proteoglycans. These are different from glycoproteins, right? Glycoprotein is the opposite. It's mainly a protein with a bit of sugar. This is a proteoglycan. So they're mainly proteoglycans. They have these GAG chains, and we have different kinds of it. That's what I want you to remember. Here we have different we have the picture to tell you that it's a diverse family of macromolecules. I don't want you to remember the individual ones. You don't need to. I'm not going to say what's the structural difference between brevican and neurocan. I'm not going to ask you that. I just included that picture just to show you that they are different. And maybe you can guess from the names that some exist in certain parts of the body versus other parts. For example, neurocan, as you can imagine, is something that would be around neurons mainly, right? And as Lamia already mentioned, proteoglycans, being part of the extracellular matrix, are very important in cellular communication and cellular migration, and in general, brain plasticity. You've also seen the ECM space in the context of synaptic transmission, right? Like every time we were talking about neurotransmitters, we said that the neurotransmitters are secreted into the extracellular space, and then they're cleared up, right? They don't, stay there for a long time. And one of the ways they get, cleared up is they get kind of sequestered by the extracellular matrix as well. For today's lecture, we're going to talk about how it's involved in brain plasticity, in pathological changes as well. But a lot of people study the ECM in the context of development and cellular migration, cell specification, etc. So we said that, again, taking a step back, we said that the ECM is mainly composed of proteoglycans and hyaluronan. And hyaluronan is, again, another glycosaminoglycan, but it is not sulfated. Okay, with proteoglycans, we said they're sulfated. So that's the difference between proteoglycans and hyaluronan. So there's this cotton candy or insulation or whatever you would like to imagine in this extracellular space. Something has to be making it, right? It's not that, oh, it was always there. No, something actively builds it and something actively destroys it. It's a dynamic entity. Just like we think of cells, always changing in response to the environment, whether it's internal or external, you have to think that if cells are changing, the extracellular space is changing as well. These are all dynamic entities. Most of it is synthesized by neurons and glia, and you already know what glial cells are. So they make the ECM, but also the ECM, the structural components of the ECM can also be broken up. And at least for the most part, a lot of people think that brain plasticity has to do with how permissive the extracellular matrix is. So if the extracellular matrix is still plastic enough, it might allow cellular or neuronal plasticity, okay? So it's almost like if you're gonna, I don't know if this is a good analogy, Anna Christina, you tell me, but you start with that whole insulation thing. Let's say you need to go and fix something in the walls. You wouldn't be able to go in and insert something between the walls if the insulation is made of cement, right? Let's say. But if it's made of a cotton candy type of insulator that's easy to move around, you can have some plasticity. You can insert something, you can hide the body or whatever it is you want to do, right? So it's a similar idea here. If the ECM is too rigid, it's not really going to support neuronal plasticity. Of course, if it's too soft, that's also a problem. It's like everything else in biology. It has to be just right, that whole Goldilocks thing. Okay. Now, we said that the extracellular space needs to be permissive enough and plastic enough for any kind of plasticity, including changes in synapses, right? And in order to change the ECM and cut up the ECM, we have extracellular enzymes that do that, extracellular proteases. And the main families that I want you to remember are MMPs and Adamses. I don't know how else to say it because the Adams family is Adamses. So M stands for matrix metalloproteinase or protease. A lot of people, different people say it differently. Or Adams enzymes, Adams family enzymes. And Adams stands for a disintegrin and metalloproteinase with TSP motifs. It rolls off the tongue. But basically, these are enzyme families. We have different MMPs, for example, MMP 1, 2, 3 until 9. As they discover a new one, they name it with a new number. But basically, these are enzymes that are remodeling the matrix, right? And again, these remodeling enzymes are also made by neurons and glia, okay? Now, as we talked about during assignment number 1, a lot of our focus as scientists has been on cells. And most people have been ignoring ECM mechanisms. What are the ECM enzymes? What are the ECM ligands? What are the molecular machinery? What's the molecular machinery that's involved? So it's kind of an understudied field. And I really think, I genuinely believe that in the next few decades, we're going to have a huge boom in ECM research and really appreciating ECM as a new explanation for brain plasticity and brain computation. A lot of the studies that are carried out about the extracellular matrix are done usually during early development because that's when the ECM is most plastic, or in conditions where you have something severe, kind of like when we're talking about epigenetics, right? Again, it's studied in the context of cancer because when you have something cancerous, you really have changes in that extracellular space as well. Or if you have ischemia, because again, when you have a cutoff to the oxygen supply, you're going to affect everything. You're really going to have like a black hole in your brain. Or when you have direct damage to the brain, like traumatic brain injury or TBI, because that's going to really affect everything, including the matrix. However, the exact details of how these proteoglycans are organized, the exact spatial organization, the exact dynamics of the extracellular space are not very well understood. Also, we don't really know how they affect brain function because partly it's difficult to study the ECM space because all the tools that we've created usually target neurons, right? When you're doing electrophysiology, you're recording from a neuron. When you're doing immunohistochemistry, you're staining usually cells, and everything is kind of focused on that. So it's a little tricky to study something for which we have limited tools. One quote I told you a couple of weeks ago was, what a very good scientist said. He said that to make a good discovery, you need a good idea and the right tool, and not always in that order. So even if people wanted to study the ECM, 30 years ago, the tools would have been extremely limited. So that's one of the reasons why it's understudied. And before I start with my talking about my research personally, I want to talk about a specific type of the ECM called the perineuronal net. And the reason I kind of want to mention it is because A, we're going to talk about it in the PowerPoint today, and B, because a lot of people study the perineuronal net because it's a little simpler to study it than just the ECM in general. Why? Because the perineuronal net is a specific type of ECM that's found around specific cells. So this is not a new thing that we just discovered. In fact, even Golgi and Ramonica Hall saw it around certain neurons. And what PNNs are specialized matrix assemblies. Okay, so they're part of the ECM, they're a specialized part of ECM, but they don't exist everywhere. They only exist around certain types of neurons. They're made up of CSPGs, or chondroitin sulfate proteoglycans, and different other ECM components. So, for example, tennyson is one, hyaluronan is another. You will see more examples in the paper I will show you later. And most often, it's found, the perineuronal net is found around inhibitory interneurons, okay? These interneurons are GABAergic, they're inhibitory, and they express parvalbumin. So usually they're around these neurons. In the past, they used to think that it was only around these inhibitory interneurons, but as time passed by, and even in my work, I found them around excitatory neurons as well. And the other thing is that if you have damage to the perineuronal net, you can have a lot of neurological disorders. So we know that during development, there's evidence from development that if you change the PNN, you're going to have a lot of issues. However, in the adult brain, the knowledge is a little bit limited. So one of the people who studies the perineuronal net in Queens College is Joshua Bromberg. Do we have anybody from the Bromberg lab here? Last year we had actually 2 people from the Bromberg lab who were working on perineuronal net projects. Do we have anybody like that this year? It seems not, or at least they're not today here today in lecture. Last year we actually had 2 people, so they were so excited to hear about perineuronal net. They're like, finally, something we already know. This is a concept you will see later in the talks. So don't worry if this is your first time hearing about it. Basically, the perineuronal net looks like something like this. So WFA stands for Wisteria floribunda agglutinin. You don't need to worry about remembering what it stands for, but basically it's something that's going to stain for the perineuronal net. And if you look at it, to you or to me, I don't know, to me it definitely looks like a little jacket around the cell, right? So what it does is that the WFA here stains the soma and the proximal dendrites. So the dendrites that are close to the soma, not the end. It doesn't, you can see it doesn't go all the way to the end of the dendrite, but the close bits of the dendrite to the soma are stained, as well as the soma itself. It's like a little jacket. And PV stands for parvalbumin. So these are the inhibitory interneurons, GABAergic interneurons that I mentioned. So you can see the soma is stained, of course. And if I superimpose, and DAPI is just a nuclear marker, it's going to mark the nucleus. And you can see, if I superimpose them, you can see this beautiful red jacket that the inhibitory interneuron in green is wearing, right? So that's what the perineuronal net looks like. It's a specialized extracellular matrix, so it's outside the cell. But you can see it's not like everywhere. It's really a jacket around the cell. It's not the cotton candy thing we talked about, okay? So we said that it's tricky to study the extracellular matrix, and that's one of the reasons why we haven't really gotten much, but we have new techniques that help out with these things that would allow us to end up staining for extracellular components. You don't need to know the details about the sushi method, but I just want you to know that it's a super resolution imaging method that is going to allow you to image the extracellular matrix. That's all I need you to know. And you can get really nice images. So this is the installation that we've been talking about. And I think this is really, really beautiful. So you can really see they look like fibers that are really kind of elongated, and it really looks like it's supporting it structurally, right? Looks like long cables going back and forth. I always find it fascinating to see the extracellular matrix. It's really a thing of beauty. And we really don't know exactly why they're, for example, arranged like this, and why isn't this fiber going that way or that way. You understand? It's super cool, and everyone is ignoring it. If this was a dendrite, everyone would say, oh, why is the dendrite connected to this? It must mean that this is where the memory is stored, or this is where. How about all these matrix fibers? They're doing something, and they're really helping this connect to that more than the others. So it's extremely important. When you see a picture like this, it really makes you wonder how anybody can ignore it. And in terms of relevance, I mean, you can argue, you can say, why would we even study the matrix? Anything we care about is localized in cells, and the matrix is not involved in anything. And you would, of course, be wrong, because the ECM is implicated in all kinds of different pathologies. For example, one of the things I studied during my PhD was disc degeneration, and we talked about the spark protein earlier in the course. And the spark protein is an extracellular matrix protein. So there's a lot of problems that can arise if you have problems with your extracellular space, including things like pulmonary fibrosis, including things like atherosclerosis, tumors, as we mentioned, but even in things that you observe very commonly, like diabetic nephropathy, that has an important ECM component as well. Now, as you know, I've been studying chronic pain for a long time. So when I was a postdoc, I was a postdoc from 2013 until 2018. So it wasn't such a long time ago, so I still remember it quite well. When I was a postdoc, one thing that always bothered me was we keep on chasing after cells in the context of chronic pain, and we keep on forgetting about the extracellular matrix. And it really was at the back of my mind almost every day when I was in the lab. And I would say, we should be looking at it. We should be looking at it. And one day I said, I'm going to write kind of a review / hypothesis type paper. And this was in 2015. And in it, I'm going to see if there's any evidence in the literature about the role of the extracellular matrix in chronic pain, and in particular, brain plasticity in the context of chronic pain. And when I did my literature review, what I found was that there were some papers showing that if you have problems with certain proteoglycans in different parts of the body, let's say in the intervertebral disc, or in the vicinity of neurons, you might have problems with myelination. You have problems with inflammation. You might have problems with neuronal guidance and migration. And also, if you have a very acute injury, for example, soon after, if you study the tissue soon after injury, you might find some problems in terms of changes in the blood-spinal cord barrier, for example, or again, myelination, inflammation, etc. You might find these things. And even in some chronic pain states, you might find that if you play with the ACM at the spinal cord level or in the periphery, you might have, again, issues with how there's involvement in how pain is ascending to the brain. However, if you look at how the brain ECM changes in response to pain, there was nothing. Even though there was evidence that the brain itself might be changing in chronic pain, there was no evidence that the extracellular matrix itself was changing. And I said, we need to investigate this. So I immediately went to my postdoc mentor's office and I said, I found a gap. I was telling you with the grant, you need to say, we know A, we know C, but there's a gap. And that's when you have the aha moment. And that's what I want to see in your specific aims page. That's exactly what happened. We're like, okay, we know that ECM is important in certain pain conditions. We know that the brain changes in response to pain, but we don't know how the ECM changes in the brain ECM changes in response to pain, and that was the gap. And when I told him that, and I had like a mini grant proposal written to show him, to kind of back up my, argument, and he said, "Oh, that makes sense. And you can investigate this if you do all the other things I had asked you to do." It's kind of like I always give the example of like Cinderella, right? Like if you sweep the chimney, you can go to the ball. And because my supervisor was not like an evil stepmother, he was a lovely man. He still is. Of course, he was true to his word. And when I was doing my projects during the day and I would have extra time, I would be looking at the brain ECM. And that's what I'm going to be showing you today. I'm going to be showing you the data that I collected, and this is already published. It was published 2 years ago, so you can see it. It's available on our lab website. Please check it out. It was published in Nature Molecular Psychiatry. So basically, what I investigated was how the hippocampal extracellular matrix changes in the context of chronic pain and pain-associated comorbidities. And as you know by now, we've already talked about it, living with chronic pain causes all kinds of comorbidities like memory problems, etc. Some of you have already seen this talk, I know, because they've perhaps they were part of the MARC program where I've given this talk, or they've attended a meeting or a colloquium where I've given this talk. So if that's the case, please bear with me. But also it's a good thing because it's something familiar, so hopefully it will be easier for you to study for today's lecture. The reason I decided, of course, to study it in the context of chronic pain as opposed to some other pathology is because, as you know, chronic pain is a very serious issue. Again, in the grant, I was telling you in your specific aims, don't just say something is a serious issue or something affects a lot of people. Give us numbers. If someone tells you chronic pain is a big deal, who cares, right? But if someone says it affects more people than cancer, heart disease, and diabetes combined, then you have their attention, right? So in your grant, remember to really show us numbers, show us statistics, show us significance, relevance. Okay. So obviously, it's a very relevant socioeconomic, ethical question that we need to address in addition to it being a medical problem. Now, why are we looking at the hippocampus? So we said we can study the brain, but why the hippocampus in general? When we're talking about pain and nociception, we talked about different areas that could be involved in the context of pain. Can anybody name one of these areas, or do you remember an area that we said was involved in chronic pain? Does anybody know? Do you remember? Well, we mentioned things like the amygdala, the prefrontal cortex. We found evidence of plasticity in those areas, but they're not the only regions. There's the anterior cingulate cortex as well, et cetera, et cetera. So there are different areas that are involved in chronic pain, including the hippocampus. But why did we decide to start with the hippocampus? Well, for many reasons. One reason, that's the real reason I think that I didn't say in the paper, but that was the real reason for me. I thought as a postdoc, I'm not an important scientist. People don't know me. If I go with something really crazy, if I study a brain area that's kind of newly implicated in chronic pain, no one is going to believe what I'm saying. It's better if I go with an area that's really studied very, very well. And the area that's studied, like I would say the most by neuroscientists in the brain is the hippocampus. Everyone loves the hippocampus. So I said, let's start with something super familiar because people are going to be more willing to accept this coming from an area like the hippocampus than a random area that no one studies. And also because we know it's involved in chronic pain. But another reason, and this is an important reason, also because, as you know, the hippocampus is involved in memory. A lot of chronic pain patients have deficits and comorbidities in all kinds of cognitive tasks. These are some quotes from actual pain patients. 15 months with no help, and I can't read anymore because I can't remember a paragraph. Imagine, like, you start reading and you don't remember what you read. Probably this is how you feel when you're cramming for an exam. But imagine if this is your entire life. It's pretty terrible. The pain is so much in my consciousness that little information can enter. I don't understand it. I used to read ravenously, and now I don't have even that for pleasure. In fact, it's causing more anxiety because I feel like I'm going senile. So you can kind of see a lot of different things here, huh? You can see that it's cognition, but it's also because you feel that your brain is working full time on processing pain, right? The pain is so much in my consciousness that little information can enter. And you also see that frustration, that something that used to be a pleasurable experience now gives you anxiety. And anxiety is another thing that chronic pain patients have. Depression, for example. Guys, I can't see your chat, so if you don't mind, can you unmute yourselves and ask? Or comment? Yes, do we have a question?
No, it wasn't faster. It's just a comment that I put there, that it's really frustrating, like how the way they feel and how it that affect and cause depression and other symptoms as well.
Yeah, it's really like everything is linked, right? And you kind of can see how it's a slippery slope. Okay? And it's almost like,
Something. So
yes.
almost every change, like connect to disease or loss of function, always cause depression and anxiety. So it's kind of too common to even think it's like connect to this memory dysfunction. That's what I think. Sorry.
Oh, but in this case, I mean, this is a quote from a patient, but there are actual studies showing that there is real memory dysfunction and cognitive dysfunction, both in humans and in animals. So something is happening that's affecting the memory. Regardless of depression, eventually it could lead to depression because you get frustrated and you get very, very depressed that you can't enjoy it. But there are tests that allow you to separate these things, and sometimes they don't happen at the same time. So, for example, depression can happen at, in mice, for example, maybe it happens at 9 months after injury, but cognitive dysfunction can start much earlier. So you can separate those things, both using behavioral testing and studying different areas that have already been shown to be involved with the emotional aspect versus the cognitive aspect versus etc. Does that make sense?
Oh, that's great. I didn't know there is a way to separate those things.
There are. It's not always easy. So that's part of the challenge, to really tease apart these things. What is coming due to the pain, just the sensory experience of pain? What is due to the cognitive dysfunction? What is due to the depression? What is due to the anxiety, right? Because a lot of the times they coexist, they're comorbid. So you have to find ways to tease them apart. So obviously one way to do it is to do it at a time point where you don't observe all of them, or to do it in patients that only show one or none, right? So some patients have pain but don't have depression, right? So there are different ways to do it, or use different types of assays, study different brain regions, etc. So when you're going down the journey of chronic pain, you end up picking up some unsavory passengers like anxiety, depression, and cognitive dysfunction. So these are things that have been shown extensively in different models as well as in humans. And of course, in humans, it's very challenging to study these things in greater detail and really tease apart the different brain regions, because you're not gonna, you're not gonna cause an injury in a human and then extract their brains. That's not how it's gonna work. You're really limited. In fact, even getting post-mortem brain tissues from pain patients is a challenge because pain is still understudied and underfunded. So tissue banks that specifically catalog and characterize chronic pain patients are very rare. That's one thing I'm always on the hunt for, because obviously this is very important for my work, and it's very difficult to get. At the end of the day, you might get one brain from a migraine patient or something, and it might be very difficult because maybe the other patient you get is a CRPS patient or a fibromyalgia patient, something that has nothing to do with the other type of pain. It's just the way they're cataloged. And of course, if you decide to go the other way, the other route, and study animals, it's a little tricky to know what they're thinking, because with humans, we can always ask them, how much pain do you have? What do you remember from this? So you're going to use a lot of behavioral assays in mice that might be difficult to design, difficult to implement, difficult to interpret, but there are ways. So for this particular paper, what we did is we caused a painful injury in the leg. You don't need to worry about the model that we use, but basically it's a painful injury that lasts for a long time. It lasts for around 20 weeks. And there are some more references for you guys if you want to know more about the specific changes that happen in the brain. But in this particular model, what we did is we caused the injury, and then we did some behavioral testing that tests both for, I'm sure you can see my cursor, you can test both for just pain, so mechanical sensitivity, as well as memory deficits. And then what we did is we labeled neurons using an adenoviral delivery method. And then we looked at how neurons change. Okay, so we did the behavior and we showed that these, again, you don't need to know details about this. I want you to understand the concepts. Please don't worry about memorizing the exact details, but understand the concepts. What you're seeing here is that the injured leg here, the ipsilateral leg to the injury, the ipsilateral foot, is obviously more sensitive. The threshold for withdrawal is lower, meaning even a small weight is going to cause the animal to withdraw its foot if you poke it with a very tiny fiber, because that paw is injured. And that makes sense, right? It's like if you have a burnt hand and someone touches it with a feather, it's not very pleasant. So this is not really surprising. The animal has an injury and therefore has pain. Okay, we created a pain model, nothing new there. But these animals also have problems with memory. For example, this is a memory of kind of remembering the location of a stranger mouse that they've met. In this case, the mouse that's being tested is a male and the stranger mouse is a female. And the male mouse encountered the female mouse, the stranger, on day one in this open field, and day two they go into an identical field, but the female is no longer there. It's instead just this open small circular cages. And what you would expect is that if the animal remembers where she was, he would go there because he would expect that she would be there, and he would investigate that part more because that's where he met her. And if an animal has an injury, it's not going to remember very well. Another assay that we do is the Y maze. Now, for the Y maze, this test relies, so you already saw the T maze, right? When we're talking about behavioral testing, we mentioned the T maze. The Y maze is similar to that. And what we did in this particular Y maze is we didn't put anything to motivate the mouse to go there in terms of a treat or a smell or anything. Instead, what we did is we relied on the animal's need for novelty, because mice are curious by nature. So if they go to arm A, next they would like to go to arm B and then C. So that would be a successful, entry sequence, because it didn't repeat itself. So the mouse, if the mouse does A and then B and then A again, that means that it forgot that it had already been in A. Okay, so this is a very commonly used assay for working memory. And what we found, of course, is that there are deficits there as well. Okay, so remember, we said that we also labeled neurons with an adenoviral delivery method. And what we found there is that if you trace the neuronal dendrites, you find differences. So this is the healthy animal and this is the injured animal. And even though the injury itself was not to the brain, you can see changes in the dendrites. The dendrites don't branch as much in the hippocampus, in hippocampal neurons. They're longer and So this is the length. They're longer and less complex. So this is a method to assess complexity. And you can see that the animals that are injured show less complexity. They branch less. This is a method known as shoal analysis. Some of you may have already heard of it, but the idea is that shoal analysis measures the complexity of the neuronal branching patterns. This took forever to do because I had to really like blind myself tracing neuronal dendrites, but eventually it was done. And then we did some electrophysiology as well to show that it's not just the looks that are changing of the neurons, but there's also a change in the plasticity levels of the neuron. In fact, we found changes in long-term potentiation. Does anybody remember what we discussed about long-term potentiation? We're talking about learning and memory.
Yeah, it's really important for learning new information.
Yes, how? How does, like, what, why, and how? So what's happening when we're causing long-term potentiation? What is this about? Nobody remembers? So, yes?
Oh,
Was that-
I think it was, I don't know if anyone else is talking. That's the whole like neurons that wire together, fire together principle, right?
Exactly. It's the molecular or cellular mechanism of that colloquial saying, right? Neurons that fire together, wire together. Very good. But if you remember, the actual experiment we talked about was the initial Bliss and Lomo experiment from the 70s in the brains of rabbits, right? Where you induce a tetanic stimulation, and then the potential stays high up for a long time, right? Days, months, maybe years, we don't know, because we're limited with how long we can keep the slice alive in our preparation. But basically the idea is that if you're causing this tetanic stimulation, you're kind of causing this potentiation that lasts for a long time. That's why it's called long-term potentiation, or LTP. So we tried to do that just to get a measure of how plastic our hippocampus is electrophysiologically. And we found increased LTP in the injured group. This is in fact the injured group, and this is the control group, the pale one. So what we found is that there's more LTP, it's more kind of primed for plasticity. And some of you may have the question saying, okay, hold on. We think that LTP usually is associated with better memory. Did you guys think of that? But these mice have worse memories. What's going on? Well, we simply don't know. LTP is not always, in fact, associated with better memory. That was the initial assumption. That's point number one. Point number two is memory for what? We did a couple of essays, and these mice did not remember where they saw a female, or did not remember which arm of the maze they entered. That does not mean they have cognitive impairment in everything. In fact, in a separate paper that I don't show here, what I showed is that injured mice have better memory than normal mice when it comes to remembering unpleasant stimuli, like unpleasant smells, which have nothing to do with the injured foot. It's a smell, it's a different modality altogether, which was very interesting to me because it just tells you how perhaps you're primed to remember bad things, right, if you have a chronic injury. So what we're showing here in terms of increased LTP doesn't mean that, you know, these mice are just smarter all of a sudden. No, it just means that it's primed for more plasticity. And this was something that was observed, so you can see something similar, increased potentiation, in a different model of chronic pain as well, which is irritable bowel syndrome, which is very painful. Okay, so today's lecture is supposed to be back. We have to go back to the extracellular matrix. Why am I talking about neurons? Well, why would you care about the extracellular matrix if nothing in the cells are changing? So the reason I wanted to first focus on the cells is I wanted to convince you that there's cellular plasticity, and that was the reason why we thought that there could be extracellular plasticity, right? Who was it who said, I think Terry Pratchett, the author, said, I'd be more inclined to see what's outside the box if I was convinced there was something in the box, or something like that. So it's like, you have to give me a reason to look in the extracellular space. If there's nothing changing in the cells, why would I care? So, as we said, extracellular matrix is studied usually in development, in geriatric conditions, in some studies looking at intense exercise, in fact, and in certain types of injury, for example, injury to the spinal cord, etc. But in our work, my thinking was that anywhere I see neuronal plasticity, I will look for extracellular plasticity. So I convinced you that there's neuronal plasticity in terms of morphology, the dendritic changes, and electrophysiology. So if the cells are changing, the extracellular matrix has to change as well. And that's when the technical difficulties began and took a lot of time to solve, because the extracellular space is extremely difficult to study. One of the things is that we talked about is that it's made up of mainly proteoglycans, and a lot of proteoglycans are very difficult to dissolve. They're insoluble. So if you want to extract the extracellular matrix so you can study it in a tube using biochemical methods, it's going to be extremely difficult. So I thought about it, and then I said, wait a minute, if I cannot extract the matrix, I can do the opposite. I can remove the cells, so I can slice up my hippocampus that has the matrix and the cells, neurons, glia, whatever, and then I can remove the cells. I can decellularize this tissue, which is exactly what I did. After a lot of trials and errors, I succeeded in removing all the cells and keeping only this matrix, all these networks of fibers, etc., all these structural components. And then I said, now that I have this matrix that's not adulterated, it's just the pure matrix itself, I can look at it, I can put it under an electron microscope, and I can see what it looks like, okay? Another thing I decided to do is I said I want to understand its biophysical properties. So this was something that wasn't really done in my field, but people in the physics department did all the time. And this was using an atomic force microscope. As the name suggests, this is something that gives you the rigidity of any object at the atomic level. And the way it's working, of course, technically, in theory, it can do that. Technically, it has many limitations, and we're not looking at the single atom resolution by any means. But the principle of it is that you're literally poking the tissue at hand with something and then seeing how much movement there is, okay? And what I did is I ended up decellularizing my hippocampi from injured versus controlled mice, and then calling this guy at the physics department saying if I can perhaps use your atomic force microscope and measure rigidity, and he said sure thing. And that's what we did. We did electron microscopy to see how it looks. And this was very shocking when we first saw what it looks like under the atomic force microscope. In the control group, we had this healthy meshwork of fibers, but in the injured group we noticed a significant thinning of fibers, okay, and elongation of fibers. So you can see the fiber diameter is really thin, and the way we did it is we went and looked at it at even higher resolution and literally measured the diameter of each fiber, which was extremely painful. Another thing we did is we used an engineering software that takes images and divides them into different describes them in terms of different geometrical motifs, just to say that the organization of the fiber is changing, because just knowing that is thinning might not be very helpful. And in terms of putting it under atomic force microscope, what we noticed, and this might not seem very impressive, one is less than the other, what does that mean? What it means, in fact, is that the extracellular matrix is now soft, okay? It's not as rigid as it's supposed to be. And that's a problem. Why is that a problem? Because the extracellular matrix has to be a certain rigidity in order for neurons to be able to branch in a healthy manner. What we found is that if you have an extracellular matrix that's extremely dense, or not dense, extremely rigid, okay, for example, 3000 pascals, or extremely soft, like 300 pascals, you're going to have problems in branching, and it needs that perfect mid-level rigidity. So in our brains, our mouse brains that we studied, we saw something that was similar to this, which is similar to what we saw, the elongated dendrites. And these are biophysical measurements that we could carry out, but we were still interested in the exact components of the matrix that are changing. So what we did is we still carried out some biochemistry of the components that we could extract. And what we found is that there were a lot of components that were found to be decreased in the injured group. For example, haplink 1. This is another link component. Remember, we said the N link and the O link. Haplink 1, agrican is a proteoglycan that's very important. This is an enzyme that synthesizes hyaluronan. It stands for hyaluronan synthase 2. Basically, these are all either components of the matrix or enzymes that make the matrix. And you can see they're all decreased in the context of injury. And at the same time, enzymes that cut up the matrix, like matrix metalloprotease 8, is increased. So there's more cutting and less building of the matrix. And finally, TIM2, tissue inhibitor of matrix metalloproteases, which inhibits the metalloprotease, is decreased. So you have more cutting of the matrix, you have less of the inhibition of the cutting, you have less synthesis of the matrix and therefore less component of the matrix, okay? So from a biochemical perspective, that was the picture that emerged. So for us, the next step was to really show that the extracellular matrix itself is regulating neuronal plasticity, because otherwise this is just a coincidence, right? Correlation is not causation. If you have changes in the neuron and changes in the matrix, it doesn't mean the matrix is changing the neuron. So we really wanted to address that. So what we did is we used a technique that parts of this technique you've already seen before, but basically what we did is that this time we used a lentivirus as opposed to an adenovirus. And we used a short hairpin RNA in order to inhibit matrix metalloprotease 8. So remember, MMP8 is what's cutting up and destroying the matrix and probably thinning out the fibers. So if we can inhibit that in a specific part of the brain using stereotactic injections, so we really drilled a hole, we did this whole surgery, we went and we injected only in the hippocampus, okay? And what we did is we inhibited MMPs in only the hippocampus after the injury was induced. And we tested for all these behaviors again to see if we can reverse these pain behaviors. So this image kind of shows us that when we do the treatment with our shRNA, indeed we have a decrease in the level of MMP, and as you can see, it's quite dramatic. Decrease in MMP because you're really stopping it altogether with your short hairpin RNA. And we did the tests again. We tested for pain and we tested for long and short-term or rather working memory. And what we found is that you can really rescue these phenotypes. Pain is not as severe once MMP8 is gone, and memory deficits are fixed as well, which makes us think that the ECM is
Dr. Tajarian? We can't hear you if you can hear us. Like, we can't hear anything you're saying.
Dr. Tajirian, could you hear us? We can't hear you. Or do you even see us? We weren't able to hear like the last 12 minutes, or what was it? 3 minutes, 5 minutes?
Did she leave? I think she left.
Yeah, she's probably restarting.
Thanks for contacting her, Sophia. I don't know, like we would not have been able to get through to her without you.
Well, I mean, I called her and I texted and like she hasn't responded. So, I mean,
I'm
they're just gonna
sure.
be like, why are you blowing up our phone? I'm like, I'm sorry, Dr. Tajirian.
Hello, everybody. It seems that
Okay,
Blackboard
you're back.
kicked me out.
Hello.
Oh my gosh, how long have this been
So,
going on?
You were going
I've
over
been talking
therapeutic
to myself.
targets. You were going over therapeutic targets when you cut out. So like
Oh,
several
okay, that's
minutes.
the last slide. Okay,
Yeah.
that's good. Oof.
Sorry, I
I
called
was
you.
like,
I
how
think
long have I been going on
just
for?
ignore that.
That's so funny. Okay, I don't know why every now and then Blackboard just kicks me out. I don't know if it happens with other people, but with me it happens quite often. All of a sudden, it's like Blackboard disconnected you. Yes. Oh my gosh, I feel so important, and people are celebrating my arrival. Okay, let me share the last screen one more time, people. I apologize for that. Okay. Alrighty, so is this what we were talking about when I
It
got
was
cut
the
off?
therapeutic technique targets.
From
So
the
we were over therapeutic.
No, I think it's 31 or 30.
Oh
Yeah,
my
it starts
gosh,
from
so it's I've
31.
been talking for a while by myself. Okay.
Yeah.
So could it have been this?
You had just explained the ECM plasticity, and you were going over therapeutic targets.
Oh my gosh. Okay. Um, so not therapeutic targets, you mean mechanism?
Well, you're
You had
talking about how
mentioned,
the ECM might be involved, and then you cut out.
You
Okay,
said that
did you
the MMPA was
hear
going
about
to
the
have,
short
and then when they,
hairpin RNA? Did you hear that?
Can you repeat that? What was that?
Did you hear about the short hairpin RNA stuff that I talked about?
No.
you
Oh,
told
okay, so alrighty, so let me go over it then. Okay, so we said that the matrix is changing in terms of biophysics. It's becoming less rigid, and the fibers are organized differently. That's what we said. And if you look at components of the matrix, they're changing too, in terms of favoring more destruction of the matrix and less synthesis of the matrix. Okay? And we said that in order to really show that this is not just a random coincidence, in fact, it's not just a random correlation and rather a causation, we wanted to see if we fix the matrix, we can fix all the other things that are happening. Can you hear me now?
Yes,
Yeah,
we
we can hear
can.
you.
Okay, good, good. Okay, so what we did there is we remember we said one of the reasons the matrix is being destroyed is because we have an elevation in an enzyme that destroys the matrix, and that enzyme is matrix metalloprotease 8. Okay, we talked about MMPs before. This is a specific MMP known as MMP8. So what we did is we wanted to be very specific. We didn't want to downregulate MMP everywhere, MMP8 everywhere in the body, because that's not a good approach. What we wanted to do is only deliver, using a lentivirus, we delivered short hairpin RNA only in the hippocampus of the mice, nowhere else. So this is the stereotactic method. You go in and you deliver this lentivirus, meaning only cells in the hippocampus are gonna not have MMP8. And then we tried to do all the testing again. And what we found is that, first of all, if you use this method, indeed you get rid of MMP8. This is after the treatment, after the shRNA. So we're succeeding. We're putting it in the right place. It's working. And if you go back and do the test, you will see that you can rescue these phenotypes. You can rescue the pain. The thresholds go up after treatment. You can rescue the memory deficits, both in terms of long-term and working memory assays that we did. So you're really improving everything if you improve the matrix. How does it work? What's the mechanism? How is fixing the matrix fixing the behavior? Well, one thing we thought about is that when we don't have a healthy matrix, we have abnormal physiology in neurons. And perhaps one specific type of matrix that's changing is a specialized matrix known as the perineuronal net. And the perineuronal net, as I told you earlier, mainly surrounds inhibitory interneurons and stabilizes them. So if we have damage to the jacket that the inhibitory interneuron wears, and now the inhibitory drive is not functioning normally, you're going to have an imbalance. You're going to have more excitation than inhibition because your inhibition is affected by this loss of perineuronal net. So the reason we thought this could be a viable approach is because some of the components of the perineuronal net, like aggrecan, we had already shown biochemically to be decreased. Okay? Oh, guys, I just realized, do you think it's still recording or did it stop recording because, oh, it's still recording. Good. Oh, good. Good, good, good. Alrighty. So that's great. We said that we already have suspicion that the perineuronal net is affected because aggrecan goes down, and aggrecan is the component of the perineuronal net. And the changes that we saw that were most notable were in a special part of the hippocampus known as the dentate gyrus. We studied all parts of the hippocampus. If you remember the circuitry of the hippocampus and the microanatomy, the dentate gyrus is this bit here. That's where we saw the most differences. In fact, the differences were quite specific. So it's not that we had an abnormality between control and injured in how much perineuronal net we had in general. In fact, that wasn't an issue. We had an increase after we stopped the cutting up of the matrix, which is normal, but starting off between the two conditions, we didn't have a significant difference. We also didn't have a significant difference in the number of inhibitory interneurons in total, but where we saw a difference was in the number of inhibitory interneurons that had the jacket, that had the perineuronal net. So in a healthy brain, healthy hippocampal dentate gyrus, we had a certain number of inhibitory interneurons that were stable, but after injury, that number dropped. And if we did our MMP8 shRNA treatment, we could reverse these abnormal changes. And if we go back and look at electrophysiology, remember we had this abnormal long-term potentiation here? We could bring it way back down to normal. This is the graphic for that. So we had abnormal potentiation, but we could bring it back to normal once we fixed the matrix metalloprotease imbalance. So in conclusion, what I want you to take home from this is that, of course, brain plasticity can have very significant extracellular mechanisms that we can study and understand, and these are really understudied. So this is really supposed to nudge you in that direction to really think about outside the box and outside the cell. And also, if you're thinking about a drug for a certain pathology, don't limit yourself to just the cells, because sometimes maybe you need to change the matrix where the cells live in. So maybe you don't need to worry about having a drug that can penetrate the cell and act there. Maybe all you need to do is have a drug that just stays in the extracellular space and does its job there. So it really opens up the door to a lot of new different venues. And in fact, when I first was doing this work, there was so much skepticism. Every time I went to a meeting, people didn't believe this data because it was so novel. And nowadays, it's one of the main things we do in our lab because, as I said, it's understudied, and I do believe that in the next couple of decades, it's going to pave the way to a new type of neuroscience and a new understanding in how the brain communicates and how the brain stores information and computes things. I know we went over the time limit because we had to repeat some things. Do you have any questions before I let you go?
I have a question, but it's more about like the research process than about the
That's fine.
reports themselves.
Sure.
I noticed that you had methods from like biochem and physics. So I'm wondering, did you get these ideas from like your own literature searches, or did you have like a team of people and you were collaborating with them?
So for this particular project, as I said, I was a postdoc, and I was a postdoc in a lab that was, so this was at Stanford, but my lab was not on the main campus. It was actually at the Veterans Affairs Hospital that was like a 7-minute drive away from the beautiful main campus that you see on the brochures. So the main campus people had the fancy fountains and the beautiful food and everything. And I was at the Veterans Affairs Hospital with hospital coffee. Very different experiences, but that's not relevant to the question. What is relevant to the question is that we, in our lab, I had an MD, PhD supervisor, so everything was very clinically oriented, and he was a clinician as well. So he did not have any graduate students or any undergraduate students for that matter. He didn't supervise students. He only had postdocs and research scientists. So when I was there, I was one of the most junior people there. Everyone else was there for like 20 years. So I didn't have a team that was working for me. I was the young one there. So I worked with teams on other projects, but this idea was mine. I came up with it. So because I was the junior people, I didn't go and recruit senior people to help me. But at the same time, there was a postgraduate student from another university who had heard about my previous publications from McGill and said he wanted to assist me, come and serve as my assistant in the lab. And he was absolutely wonderful. So he started helping me with some of this work later. So it was me with the assistance of Victor. However, most research is not done like that. My other projects in the lab were done within teams. And in a lot of research labs that have students there, usually it's a group of students working together. But at that point, we didn't. It was just a different structure to the lab. If I were a graduate student and had I come up with this idea, it would have been different. Graduate students don't have the same independence as postdocs. You know, I was already a senior postdoc. I had published many papers with my PI. He trusted me. And he's a nice guy because, I mean, there are many PIs who are a lot more, restrictive and perhaps not as permissive as Dave is. So I just happened to have a very positive experience there. So I was allowed to carry this on as if it was my lab, and I brought it here with me in my own lab now. So does that answer your question?
Yeah, like I was just wondering, I guess more like how you got the idea to bring in these like cross-disciplinary
Oh, why did I think about? Okay. Do you want the real answer to that? And you promised you won't lose faith in me. And this applies to everybody. The real reason for that is that when I was an undergrad, I did not pay too much attention in class. That's the real reason. And what do I mean by that is that I didn't limit myself to what I heard in class, because I would hear things in class and then I would go and research it by myself. So as you can imagine, I didn't get A's in that class because I didn't focus on what the teacher was asking me to focus on. Instead, I just was like all over the place. So I kept a very open mind in terms of what could be possible. So the teacher would say, oh, A goes to B goes to C. I would see it and say, oh, okay, whatever. I would forget A goes to B goes to C. I would say, where can A possibly go? And look that up. and prepared for the exam. And the teacher would say, where does A go to? And I wouldn't say B, I would say it could go to D, it could go to F, and I would get a 0 on that question. So it wasn't ideal for getting good grades in undergrad, but it served me, that kind of lack of specific knowledge served me so well as a researcher, because I went to it like an idiotic child who knows nothing. Literally, I went to it and I said, oh, how am I going to study this? Other people haven't studied it. Why haven't other people studied it? Is it because I'm much dumber than them or much smarter, or is there another problem? And usually it's neither. Usually you're not very dumb or very smart. Usually there's another problem. And the other problem is that there's a lot of technical limitations. And I said, oh, what's the technical limitations? Oh, it's insoluble. Okay, then if I cannot remove the ECM components, what else can I do? Maybe I can remove everything else. Is there a method that does that? Let me research it. Oh, someone in France did this in a different type of tissue. Can I adapt it to the brain? Let me try it. Victor, do you have a minute? Come help me here. So we did that. And it was really like that, step by step. And then I had the matrix in my hands. I'm like, okay, great. Now I have a matrix that's been decellularized. How can I study it? And I said, okay, what does a child do? You know, if you see something, you don't know what it is, you look at it and you poke at it. And literally that's what we did in fancy ways. Instead of looking at it with your eyes, we put it under an electron microscope. Instead of poking at it with your hand or with a stick, you end up poking at it with this tip of a cantilever with the thing, with an atomic force microscope. And I think one of the things that helped is literally I just ended up picking up the phone and asking, I want to do this. It may seem crazy to you, but I want to do it. You know, can I come and use your machine? And at the end of the day, the worst thing that will happen is that it will go a different way and whatever. If you're good with time and you're still carrying out your main duties and this is something you're doing on the side, why not? Why not try new things, right? Because that's how things advance. If you stick too much with what is already known, you're not going to discover something new. And again, maybe you've noticed that's how I teach this course too. I don't ask you to memorize a lot of details because, truth be told, a lot of these details can be disproven tomorrow, or maybe were already disproven yesterday and we don't know about it. So there's no point in you going to graduate school thinking that A definitely goes for B, to B. I want you instead to learn that there's some evidence that A goes to B with the following limitations, because these were the tools they used. That's what I want people to understand. And I think if you've learned that, I've succeeded in my mission, because the rest is, you know, it's just Jeopardy information. You can memorize it, you can refer to a book, but it's not going to help you as scientists. You're very welcome. Okay, guys, just in the interest of time, I might let you go because I have a few people who want to meet with me. So if you're not scheduled to meet with me after class, I will see you next Monday for our last week of classes. Unfortunately, it really, like the last bit really flew by. Have a nice day, everybody. See you on Monday. And if you're meeting with me, please stick around. So, okay, so we still have too many people. Let's wait for the crowds to thin out, and we'll keep the people who had asked for an appointment earlier on. Okay, perfect. So these are the three people we have, Sophia, Noor, and Roberta. Now, if you have a question that you don't want other people to hear, do you mind coming back in 5 minutes?
Yeah, do you also want to stop recording, Dr. Zarian?
Oh, absolutely. Thank you for reminding me of that.